So good morning again, and welcome to lecture 4. In this lecture, what we're going to do is we're going to take a look at how other bacteria or other different source of ways of bacteria to get energy. Because last lecture we focused on how we can use glucose, how bacteria could use glucose to get energy through glycolysis or TCA cycle to generate either 2 ATP or 38 ATPs as energy source. And in the process, by breaking down the glucose into intermediates, and those intermediates then can be used to make DNA, amino acids, and all sorts of molecules, right? But what happens when you don't have glucose or you don't have oxygen? Okay, and so this lecture is to really look at a diverse approach that microbes use to generate energy, because the living environment may not have glucose, may not have oxygen, and then what? Life always find a way, and that's one of the, if you remember from Jurassic Park, the geneticist was saying, well, they claim the, I don't know, is it too old for you guys, the Jurassic Park movie? The guy claimed they built it. They claim they built a barrier so the dinosaurs won't break the barrier. But the geneticist says no, life will find a way, and that's always true. This is the theme of this particular lecture. We'll say, how do other microbes find a way to make a good living and live happily ever after? Okay, so we're going to take a look at that. But the first thing is I wanted to review a quick highlight that the glucose fermentation, that's the one that we spend most time talking about. That's most represented by the yeast. Okay, yeast is doing the fermentation, as we say. We know them well. We use yeast to make wine and to make beer, and these things happens automatically, or not automatically, but in nature. So if you a fruit comes on trees and falls onto the ground, and they will be fermented. And if any animal picks up and will eat it, and they get drunk. If you haven't seen it before, elephants, when they pick up fruit or apples falling down a tree and ferment it, they get drunk. Now, you can imagine what will happen if you see elephant gets drunk. And look up the video. It's really funny. All right. So that's fermentation, yeast, and then the glucose respiration, the respiration we talked about, is that they break down the glucose and goes through TCA cycle, generate NADH and FDAH. And by now, I assume you have calculated, you understand where does that 38 ATP comes from. But this all depends on this process can keep going, is because you have a electron gets passed along, and you can dump the electron somewhere, right? In this case, electron goes to the electron receptor is the oxygen. So oxygen takes the electron and becomes what? Water, right? Now, and so, but what happens if you don't have oxygen? Well, if we don't have oxygen, we die, right? Because we live in environment. But can E. coli still do glucose respiration, but without oxygen? Okay, so that's it, that process. The short answer is yes, and the process is called anaerobic respiration, compared to aerobic respiration, meaning anaerobic is a process breathed without oxygen. Now you will find, you'll soon wish that we have that ability. Then we can go into water, and we don't have to depend on the oxygen, right? And we can go to Mars. We don't have to use the oxygen. So this is, let me quickly review. This is, you already know very well, this is aerobic respiration. It's a different presentation. So NADH donates the electron, and hold on. Yes. So the question was E. coli. Yes, they can. In the absence of oxygen, they can generate 2 ATPs. So but is there any way they can generate more ATP than 2? Okay. So let's first take a look at the aerobic respiration. So you have, let's see if I can. So the electron floats and floats, and then comes here to gives the oxygen and proton, and then you got water. Okay. Now in the process, the proton gets pumped out. All right. So then the question is, what if you don't have oxygen? Then what? And now you can so if you look at in comparison this side by side, and now you can take a look at this one. Hold on 1 second. So right, this is nitrate, and nitrate can take on electron and proton. Okay, and become nitrite, and nitrite can diffuse out of the cell. Okay, so the nitrate can go can be reduced, can function as electron receptor, okay, and then be nitrite. And so now the electron, the key here is then the electron can go, right, and then the NADH can return to become NAD, and then you get NAD can go to the TCA cycle to bring more electron. So now the process is can still go, and so the electron transport chain can still run, and then the difference though is this one. So if you look at this, they're using a different, instead of cytochrome O, and they use a nitrate reductase, an enzyme. Now because of this, and the difference between these two is the proton are 2 proton less. Okay. Now, if you have 2 proton less, that means you add it up because 3 proton is required to make one, one ATP, right? And so with 8, you have almost 3 ATPs. I'm sorry, with 10, you have 3 point something ATP. But with this 2, you have 2 and ATP, right? So if you have to multiply more, right? And so then you do the calculation, then you can see this anaerobic respiration is less efficient than the aerobic, but it is still pumps out the proton, right? A little bit less. Okay, so nitrate is actually pretty efficient in taking out the in receiving the electron and keep it going. All right, and so the there are other so here's the nitrate. Now this is where the nitrate is. Okay, so the nitrate goes to nitride, and then this can support the respiration, anaerobic respiration. So this is called the nitrate respiration. And if you notice, this is aerobic respiration, and you can actually have ion respiration. And ion respiration is even more efficient than the nitrate. And then going towards less efficient, so these numbers, the electron potential, the smaller, the less efficient. And so this actually, the sulfate respiration is also very common. Okay, so these are all common electron receptors that can support anaerobic respiration. Okay. Now, and including this, wouldn't you be, wouldn't it be wonderful if we can actually use CO2 as the electron receptor? Because we just gave out the CO2, right? There are other microbes that can use CO2 as an electron receptor and make and extract energy. Okay. So I like to highlight that from a sulfuric acid. Okay, you can, and then you have sulfur, and then you have hydrogen sulfide. And so those are 3 different molecules. We'll come back to talk about the in detail about the reductions. And so here are examples of from sulfuric acid. Okay. So here's sulfate, and I'm sorry, this is nitrate, nitride, and these are different steps of oxidization. And then E. coli has the nitrate reductase. So E. coli can carry out this particular step. Okay, but E. coli will not be able to use nitride as a reductase. However, if you look at the Pseudomonas stazeri, and this is a bacteria that has all these enzymes. Okay, depends on whatever the steps you have, and they can oxidize all of it, all the way to nitrogen. Okay, now nitrogen is a very stable molecule. It has 3 bonds between them, covalent bonds between the 2 nitrogen molecule, right? And so and it is completely reduced. So there's no very stable. Okay. And then you can look at the others. This is a sulfuric acid, right? And so then you can have sulfurase that can reduce, and this is sulfide, and then you have hydrogen sulfide, and then these can be excreted. And so a different approach is through this process called the assimilative sulfate reduction. The key here for us, for microbiologists, what we care about is this one. You're going to hear me talking about this again and again. So from sulfate to hydrogen sulfide, you that absorbs 6 electrons. I mean 8. I'm sorry, 2 and 6. And so this molecule, if you completely oxidize, so this process is reduction, right? From this direction here, this is reduction, and then the reverse, it is oxidation. How do you I know it's hard, sometimes you try to memorize which direction is oxidase, which direction is reduction. Well, oxidation is easier to remember. You see there's no oxygen here, right? Well, this has oxygen, this has oxygen. So if you're going the other direction, you're adding oxygen into a molecule. Well, yeah, that's an oxidation. Okay, that's how that may help you remember which one. I'll keep it straight, which one is which. All right, and hopefully this is helpful. But the key here is the electrons, all right, 8 electrons altogether from sulfate to hydrogen sulfide. All right, we'll come back to this very important question later. So that's about the anaerobic respiration. Now, the point is earlier, one of you asked, so, well, can E. coli does fermentation? Yes, E. coli is very, very versatile. It's a facultative anaerobe, and basically it does anaerobic respiration, fermentation, aerobic respiration. But we're back to the choice. If E. coli, what choice would E. coli choose? If E. coli has all 3 processes capable, then under what condition it will do what? What would be the standard? Yes, Sean, go ahead.
If there's oxygen present and it's a fair amount, it would likely do aerobic respiration. And then if there was less oxygen, it would pick the anaerobic respiration. And if there's no oxygen, probably fermentation.
Yeah. So, but I wanted to be very picky about your answer. Is actually it has to be no oxygen. If there's oxygen, any oxygen left, it will still do respiration. And then if there's absolutely no oxygen left, and then the E. coli will look around and see that table. Remember that table? Now this table. Now, yes, we can look at the table and say, okay, what do I have, right? But E. coli can't. But E. coli looks around in the dark and feel, I don't have oxygen. Do I have iron, ferric? Do I have nitrate? And then they go down the list, right? And if it doesn't have any of these, and say, I'm doomed, then I'm going to go to fermentation because that still gives me 2 ATPs. It's not preferable. So oxygen gives you 38 ATP, and then everybody between will give you less than 38, but still more than 2 ATPs, right? Okay, so that's how it will choose, right? And so, but in nature, so these are the model organisms that I hope that by this discussion that further clarified all these approaches. So now we're going to look at a diverse array of bacteria. And so the first thing we want to talk about is the purple sulfur bacteria. Now we talked about bacteria have color, and we talked about bacteria being energy conservative, and they won't waste anything if they won't produce anything if they don't absolutely need it, right? And so this bacteria is purple. And so the question is, why is purple? Well, we earlier always said, well, purple was because it's a photosynthesis bacteria. It does photosynthesis. So we're going to first look at its energy source, energy, and then we'll come back to look at the CO2 and the reducing power, what I call reducing power. Okay, so the light, now how do purple sulfur bacteria get energy from light? It has a photosynthesis center. It can receive the red or infrared light, and then this photosynthesis converts the energy received from light and gets into a high energy molecule. So you have low energy and high energy, and then the energy passes from all these molecules and then comes back. The key here is the electron. The energy, it energizes, and then this converts into high energy electron, and this molecule passes the electron to these molecules, electron transfer chain essentially, and then becomes the low energy molecule again. So in the process, the electrons are circulating. Okay, the electron doesn't go out, but obviously there are some electrons needs to sometimes intermediates gets used, so you do have to have a supply of electrons. But in general, the electron this is a closed circuit. The electron just floats, and in the process, it generates a proton gradient across the membrane. Okay, so let's take a look, and this graph shows you what it does. Okay, and so here's the light. So here's the light. All right, and the light comes here and generates and this gets the energy and pushes the electron and goes in circles. And in process, 2 protons gets pumped out. And you say, oh my God, this is a very, very inefficient process, right? When NADH can pump out 10 protons. Well, but the good thing is What's the limit of light? As long as it's exposed to the light, and it just keeps going, right? And also, you can have more of these photosynthesis centers, right? And you can also have, these are called the light harvesting center, which I'll show you later. But so in the process, you can generate a lot of proton, and then the proton can come back in and generate the ATP. Okay, so still 3 protons generates 1 ATP. And the light harvesting center, this is how it works. This is almost like a satellite. The best way you think about this is a satellite dish, right? It's a reflective curvature and reflects all the focuses all the light into that one sensor in the middle, and that concentrates the light, right? And now there's another thing. We know trees are green and the bacteria is purple. And why is green? Why is purple? Well, these are the molecule that absorb the light. And okay, so this is the bacterial chlorophyll, and this is the regular chlorophyll. And so these, if you look at this, the red light, the green, the green, and the green is telling you what light it is observing. So this is the observance. So the tree, or the chlorophyll in the tree, actually cyanobacteria as well, that they absorb in this range, but they don't absorb here. Okay, and this is where the green light is. Okay, so that means this. Okay, so now you understand why tree is green. This will be a good question, right? Well, why is tree green? Well, because the green light is not absorbed by tree. That's the leftover light. Okay, and so and then then you can say, well, what is why is purple bacteria purple? Well, because this is the area that's not observed. Okay, now then the question is, why do bacteria prefer a purple color? Why don't they just get the green as well? There are cyanobacteria that's a green, right? Right. How come you have cyanobacteria that's green, but you also have purple sulfur bacteria? Anyone want to take a guess? Well, I can tell you the purple sulfur bacteria, if you remember the picture that we show, it typically lives in streams, in forests. All right, and so in forests, the sun goes through the tree leaves, and then get to the forest floor, and then you look at well, the tree absorb all the light in this area, and then the bacteria needs to absorb these light. They peak here. Okay, so that's one reason. Nature, the bacteria. This is an example of bacteria find a way. Well, the bacteria can't compete with the tree because the tree is taller. Bacteria is stuck in the mud. Purple sulfur bacteria typically lives in low streams and mud, and they can compete with trees, right? So, okay, well, I won't compete for the green light. Let me absorb the purple. And I'm sorry, let me absorb the 800 and 700, the far red. And so that's why they look like purple. And so
Professor, can you go back to that slide?
Sure.
Yeah, I see that the green color. Is the green color indication of where plants, which type of light they absorb? Is it based on that?
Yes.
I see green. The wavelength is from like 500, 510, 550. So do plants absorb light at that range of based on the wavelength, or why?
Yeah. So okay. Yes. So let me how do I let me I'm getting fancy. I'm erasing all the drawings so it'll be more clear. Now, it is really important for everyone here to know and learn how to read graphs. Okay, it is absolutely essential. So what this graph on the wait, let me go to drawing. Okay, what this graph says is this is the wavelength, right? And this y-axis is the absorbance. That means the amount of light that's being absorbed. And so if you follow the green curve is for the chlorophyll, that's for cyanobacteria and then tree. And so if you follow that and you say, okay, that peaks at 430, 480, and then it's not observing at this point, 550 to 620, but it peaks at 680. Okay, so this means this curve just means the chlorophyll is not efficient at absorbing light in this region. Okay, so the tree doesn't absorb light in this region or less efficient. Is that clear?
So yeah, if I understand in this, in the wavelength based on the color below that graph, so the diagram. So I see that the green color is like from 475 to around 550. So does that mean they just coloring what like a green plant doesn't absorb?
Yeah, this is the range the green plant doesn't absorb. And the reason that the plant looks green is because they don't absorb the green light. And that's why it's visible to your eye.
Okay, I got it now. Thank you.
Okay. And the same approach applied to bacteria. All right. So now we're going to look at the structure. How do they actually absorb the light? Now we know the light needs to come from the outside, and so then these bacterial chlorophylls are in these membrane structure. And so why do they need to be in membrane? Well, because the electron transport chain is in membrane, because electron transport chain, unlike the DCA cycle, they do need to pass electron from one to the other, so they do need to be together. Okay, and so that's why you see these structures here, these membrane vesicles. And if you look them up, it's close to the cell membrane. And then you have bacterial chlorophylls in here. These are in this cell membrane. And okay, the green is chlorosome. Yeah, they call chlorosome. And there are other examples of membrane. They just have lots of membranes stacked together, and for phototrophs, and or you can have lots of membrane vesicles. Instead of long sheets of membrane, they use these vesicles, tiny vesicles, do the photosynthesis. Okay. So what we just talked about is the green and sulfur bacteria. They use light as energy and generate ATP. Okay. Well, but there are other. So this is one way, all right. But then there are other photosynthesis. So this is focused on here, right? And this is light, how light generates energy. But are there other ways of generating energy to the photosynthesis? We alluded to earlier, you have cyanobacteria, okay, algae and green plants, and they use light to generate energy. And so how do they use light to generate energy? And so let's take a look. And this is the Z scheme for photosynthesis. And the key to remember this is that this is essentially have two photosynthesis systems linked together. So you have photosynthesis 1 and photosynthesis 2. All right. Now, the photosynthesis 2, this is the wavelength. It absorbed the light, 680, and it absorbed the light and become high energy. Now, notice this. The electrons, where do they get it? So the electrons is being stripped from water. And so the water, what we know, oxygen, when oxygen use an electron receptor and to generate water, right? And so we know in this opposite direction, water, when you split water, and that will generate electrons and oxygen. And now this is extremely important to understand. Say, water is very, very stable, right? So imagine how much force, how much energy you have to put in to split the water to become oxygen and proton, and so that you can get the electron out, right? So this is a very, very energy efficient process, and this oxygen, and this is why the green plant generates oxygen. All right. Now the electron goes up and then goes down. So if you follow the electron, all right, goes up here, goes down to P700, and then it gets and it gets energized again, and then goes to ended up being NADHP. Okay, NADA, NADA, or NADPH. So the question was, where did they get the energy? Well, the energy is from the sunlight. That's just how much light how much energy is in the light. Say, well, if you want to feel how strong the light is, you can compare. You can just get a white paper and a black paper, or even a fiber, right? You get to feel if you wear black versus you wear white, how much energy difference between, black absorbs the energy, right? If you get a black or dark colored fiber, and you will feel the difference, right? That's just how much energy that's in the light. Now, for in this process, it's important to see the electrons goes all the way to NADH. Okay, it's almost a conceptually a reverse of the respiration process, right? Aerobic respiration, you take glucose and generate NADH, and the NADH donates electron and flows backwards and to the oxygen and now to water. All right. So so if you want to get that process reversed, then you have to generate lots of use a lot of energy to pull them apart. So now you understand why oxygen is a high energy molecule, right? Because you actually have to absorb the light in order to break this up to get the oxygen. Okay, so in this process, one you need to know the light, the cyanobacteria or the green plant has the Z scheme photosynthesis, has two photosynthesis centers. So that it gets energized twice with the light. And the first one, the 680, is the one that's most efficient in absorb sunlight and split water. It gets so much energy, enough to split water into oxygen and extract the electrons. And the electron goes to NADH. All right. So the NADH, well, so that's where we have here NADH. Generates the with the light. All right. Now this brings us to why do you need NADH, right? Well, the energy, the NADH, keep that in mind. The H is for reducing power. Okay, so why do we need the reducing power? We'll come back to this in a second. And so this is a spectrum of cyanobacteria where the light gets absorbed, and it requires a assistant other molecule. It's phytocyanin. It's the a molecule that helps absorb the light. Now, this we just talked about the oxygenic bacteria that generates the oxygen. The green cyanobacteria generates the oxygen in this photosynthesis process, right? And that's why they're called the oxygenic process. And so you essentially split the water and to generate the oxygen, and that creates the reducing power. Okay. Now, then the question is, well, why do you need electron? Well, electrons couple to protons, right? So the easiest way for you to remember or to keep which one is which is the carbon source. Okay, so why do you need electron? Well, we need electron because we need to make biomolecules from CO2. Okay, so in comparison, what you hear is the N-oxygenic photosynthesis. So the photosynthesis of purple sulfur, purple and the green bacteria, they do not generate oxygen, right? Why? Well, because they use the energy, the electrons are mostly circulating, but and so they in the process they don't split anything, but they still need reducing power, right? They still need that H. Remember H. And so where do they get the H? Well, they get H from here, hydrogen sulfide, and that's coupled to the electron. Okay, electron is negative, proton is positive, and so the two, yes, they get separated, but mostly when they become stable molecule, they are together. So remember, from sulfate to here, how many electrons? Earlier we talked about, it's 2. Yes, it's 2 here, 2, oh, that's horrible, and 6, right? But if you reverse that, you from here to here, you get 6 electrons, and if you go from here to here, you get 2 electrons, right? So these electrons get extracted. Okay, so then the question is, okay, now we understand you need electron, and you need a proton. So now we're going to take a look at what do you do with the electrons? Okay, so now what we just talked about is we use light to generate energy for purple and cyanobacteria. They're different in terms of reducing power. Where do they get the reducing power? The purple sulfur is from hydrogen sulfide, and this simply is from light. Actually is, or you can think about is water, right? The electrons, the source of electron, that's water. And so now if you have those, what you need is the carbon source, right? Because if you think about build a new bacteria cell, what you need is the carbon, right? Carbon source. Almost all biomolecules have carbons in them, right? And so for the unfortunate, for the fortunate bacteria like E. coli, the carbon source is glucose. But what about other bacteria like cyanobacteria or purple bacteria? Cyanobacteria lives in water. Is there glucose in the water? No, the water. And what about the mud or any other places? The glucose is very, very, very difficult to find. So actually, this would be a good place to ask a question. We said glucose is the most common common source of most common carbon source for all bacteria. Almost every bacteria can use glucose, right? So it's very common. So in theory, when you go to a mud, you should be able to see glucose, right? Because it's supposed to be very common. But we know when you go to mud, if you're brave enough to taste mud or water from a stream, would you be able to find glucose there? That's a question for the class. Anyone want to give it a try? So if you go to a stream, a spring, no, not spring, but stream water, or because that's what's in the soil, right? And say, okay, Mamadou, and you change your answer from yes to no. Why? Ha. Can you explain why do you change your answer?
We know that, like in stream, we have those cyanobacteria who perform photosynthesis. So they're doing it because they don't have glucose. If there was glucose, you mostly seen other bacteria, and they doing photosynthesis to get the glucose because they don't have it.
Yes, that is very, very correct. Now, but the question is, you still haven't answered my question is, well, if glucose is such common source, every bacteria can use, then why can you find them in nature, right? Because everyone used them, so it must be common in the nature. So it must be you should be able to find them, right? But if you taste the soil, I wouldn't claim I tasted, but if you do, if you are brave enough to taste the soil, would you be able to taste it? Would it be sweet and with glucose? It's a fun brain exercise.
I think in the presence of cyanobacteria, because they're making glucose, if you are brave enough to taste it, you will find, glucose.
Okay, so you think if you have cyanobacteria in the liquid, and if you taste what's outside the cyanobacteria, you should sense the glucose. Okay, well, anyone else wanted to give a try? Okay, so we assume, so this is an interesting question, right? Well, we assume all kids likes right? The candy right? And so, but if you go to a kindergarten class and you not dump, but if you spread the on the table, and do you think the will be there if in 10 minutes? Why not? Well, because all the kids around the table just munch it up, right? So the question about whether or not there's glucose in the mud is yes, but if you dump glucose in the mud, it's gonna just take up. Whenever there's a glucose, it gets picked up by whichever bacteria it is around. And that's the reason every bacteria can use glucose, and you can't find glucose in the nature. Everyone is competing. Okay, that's a quick diversion and break. Okay, now back to the question that's in front of us, saying, okay, now the bacteria living in nature, and say, okay, we can't find glucose, but we need a carbon source. And what do we have? Well, CO2 is all we have. What do we do? Well, remember, life finds a way. Well, that's a good example. Okay, so this is regarding to the carbon source. Okay. So most bacteria live in the soil. They depend on organic carbon source, but for Phototropes, the bacteria does photosynthesis, and the the good thing is that they have lots of energy, and then they can either use CO2 as the carbon source, or they can use organic carbon source. And so it depends on what they do, you can divide them into troops and troops. Now troops just means microbes. Plural here referring to organic, auto here referring to use CO2. It's not an automobile auto, it's just related to CO2 use. Okay, and then comes in the famous Calvin cycle. And again, this is a bunch of enzymes and a bunch of intermediates. And my connection is poor. Can anyone hear me?
Slightly come in and out.
Oh, it's choppy. Okay. Hold on a second. I don't know what is going on. Yeah, okay. So it recovers a little bit. Now it's better because I see my connection. Okay. Maybe internet needs a coffee break or something. Okay. All right. So that was my coffee break. So now come back to the Calvin cycle. The Calvin cycle, like everything else that we've talked about, is this. This is the key I care about. And so what we care about is this equation. It's 6 CO2, 12 NADPH, 18 ATP, and then you generate 1 glucose. Okay. So this is the overall equation, and we talked about the 1 glucose. If you metabolize it most efficiently, that will give you 38 ATP, right? And now, then how many ATP are needed to make 1 glucose molecule? Assuming 1 NADH is equivalent to 3 ATPs, right? That's what we calculated in the past. And so this then this gives you a calculation of 1 glucose. To make 1 glucose, you need 54 ATP, right? So it'd be 36 plus 18, be 54. And so that makes sense, right? Because you do need more more energy to make an energy, and then releases less because energy gets consumed in the process. So that just gives you a sense of how expensive to make glucose, right? So it consumes 54 ATP to make 1 glucose, which can release at most 38 ATP, right? So this is why the Calvin cycle, or the carbon fixation, can only take place in phototrophs, in a bacteria or any organism that does photosynthesis. And that's why tree can absorb CO2 and turn CO2 into glucose and cellulose and then wood, right? Because that requires energy from the sunlight. Okay, but if you think about it, all organism living on Earth requires the energy from sunlight, right? And we're getting energy from not directly from sunlight because we eat plant material, right? Because corn and grain, that's what we farm and eat, and because that's starch that's providing the energy, right? So this is the overall reaction. But then, so now we can take a look at this supposed cycle. And the cycle is comprised with multiple intermediates and the enzymes. Okay, so we'll, and this is, the start is here. This is where the goes into the CO2. And then the ATPs, NADPH, and then fructose comes out. And this is what comes out. And then this then goes into the biosynthesis. And the reason this is really complex is because there's lots of unstable intermediates, and so this is really just to tell you what CO2 goes in, carbon dioxide, and this is the intermediate ribulose biphosphate, ribulose bisulfate. And then you have unstable intermediate, and then you generate actually two. So it goes from, this is 5 carbon plus 1, and then you comes out to have 3 carbon molecules. And this is called the phosphoglyceric acid. And that goes here to generate these two intermediate molecules, and eventually comes back here to ribulose bisphosphate. And so now you have, and then the cycle repeat itself. Okay, so now it repeats again. Okay, and so this really is okay. So this is the cycle that it actually runs. Okay, with the light, and because the light, remember the light gives you this NADPH, right? That's with the electron. And the light also generates the ATP, and those are the things. And the plant is exposed, or cyanobacteria exposed to CO2 in the air, and those are the key ingredients that's required for the Calvin cycle. And this cycle is different from photosynthesis. These are the enzymes and intermediates floating in the inside the cell, cytoplasm. And again, the overall is where we are most interested in these, that goes in and what comes out. Any questions? This is probably most difficult to follow. Okay. Okay, so what we just talked about is the purple sulfur bacteria and cyanobacteria. And so now at this point, you understand the energy is the from photosynthesis, and they have two different schemes of photosynthesis, and that's related to how they get the reducing power, the NADPH, and this is essentially getting the electron, right? So the electrons, and how do they get the electron? And this is from the hydrogen sulfide, and this is from reducing power, actually the light, and then split the water, right? Again. And then these reducing power then used to fix CO2 as the carbon source. And so this is using Kelvin cycle, and that's true for both of them. And so now we're going to venture to other bacteria. And so one of the interesting bacteria is hydrogen bacteria. And hydrogen, and does hydrogen have energy? For safety reasons, these nowadays you guys don't get to play with generating hydrogen. In chemistry lab, we used to generate hydrogen and lit it on fire. But you should also know there's a hydrogen, hydrogen cars, right? Why? Well, because hydrogen and can be oxidized and in the process generate water. And you can burn hydrogen, it's a flame, right? And also hydrogen can explode, right? No, hydrogen can be oxidized. It has weaker bonds, and hydrogen is very unstable actually. And in I was partially joking because when we were doing the experiments decades ago of not recognizing how unstable it was. When you generate hydrogen, if you're not careful, when you lit it up, it can actually have small explosions because the glassware can actually explode. But back then, but the hydrogen you generate it's very small. But nowadays it's considered not to be safe, so you don't get to use. But anyhow, but hydrogen can be burned. And that's why the balloons that nowadays you use helium balloon, right? You don't use hydrogen balloon. Why? Well, because do you guys saw the airship that was burned? Historically, that's in the 30s and 40s, and they were using these giant balloon to flow as an airship. And those. Yes, the Hindenburg, that's right. They back then they will use hydrogen, right, not helium, and then they didn't realize. Well, they knew hydrogen was not safe, but they didn't have alternative to generate helium. It's more difficult to generate. Anyhow, so just all this is just to tell you, hydrogen is very has a lot of energy and can generate. You can see this is how much energy it can generate. All right, kilojoules of energy. And so in this process, then the hydrogen and oxygen and CO2 together, they can generate methane or molecules. And so this is an example of hydrogen molecule can be used to generate energy. And so this bacteria called Ralstonia, is hydrogen bacteria. It just solely grow on hydrogen. Now in nature, you will only find this bacteria where there's a hydrogen source, right? You wouldn't find this bacteria in your backyard because there's no hydrogen. It can't grow. So this bacteria actually use two hydrogen enzymes. So membrane integrated hydrogenase. And so it splits the hydrogen into proton, and then and the electron gets transferred to oxygen and generate water. Okay. In the process, the proton gets pumped out and that generates a proton gradient. All right. Now in here, it also uses CO2 and the I'm sorry, inside it has another hydrogenase. It splits the hydrogen into proton and the and electrons, and then charged NADH. And we know the reducing power is needed to fix CO2 to make cell material. Okay, so that is how this hydrogen bacteria gets energy and to grow, because and you can see it needs lots of hydrogen to grow, right? Because otherwise it won't be able to grow. So that's a good example of hydrogen bacteria. And then you have methanogens, and this bacteria also uses the hydrogen as the energy source and the reducing power, but in the process it generates I'm sorry. So this is the Methanogenesis. It generates it generates methane here. Starts with the hydrogen as the energy source, and it depends on. So in this case, it uses the inorganic. It uses inorganic carbon as the source for carbon source. Okay. Now this is a graph. I put it up. I included and purposefully to get you completely lost in the process. No, I'm just kidding. I'm just trying to impress you on how complex this process is. And what I liked you to circle is here. The CO2 is in, the hydrogen is in, where the hydrogen comes in, it provides the energy. And the methane is generated. All right. This is, and then graphically, how this is, you actually see this is in the process, is that the hydrogen, not surprisingly, is here. Okay. And gets split and to generate proton gradient, and and then the electrons are actually involved in making these intermediate molecules that's for fixing CO2 to make methane, okay, in the methanogenesis process. Okay, so you would think the methane is a waste, well, in this case, it's a waste product, right, methane, because the bacteria is getting energy from hydrogen, and then it generates methane as the end product. All right, and so you see this is so this is methanogens, and then another bacteria that's quite confusing to most people is that you can actually have methanol troops. So methanogene actually generates methane, and methanol troops is actually using methane as an energy source. Okay, for everything, energy and carbon and reducing power. It uses methane exclusively for everything. That's methanol troops. So here is an example of how methane is being used in this process. So methane gets oxidized. Now, well, okay, and so now this is enzyme. Then you have methanol, and then you get further further oxidation, and so lost another hydrogen, and then this gets further oxidized, and so NADH are generated, and CO2 is released. And okay, and so these then this is going to biosynthesis, right? This is the common source. Now in this here, then the outside, then the proton gets the NADH comes here and donates donates electron because NADH had to come here. NAD has to come here, right? All these NADH had to be recycled. And where does it recycle goes? Well, NADH comes here, right? And goes from here to here, right? And in the process, the proton gets pumped out, and the electron goes all the way here to oxygen, and you need oxygen for the methanol troops. And now and then the proton gradient comes in here and generates ATP, right? So now you have ATP, and you have energy source, and then you also have carbon source. That's all you need for the bacteria to grow. All right. So if you're still not sure about this process, is think about methane. It's natural gas, right? And that's where you can use to cooking, right? Then you burn it with oxygen, and you generate CO2, and that's where you get the energy source. Okay, so so I think so. Are there any questions about all this we've just talked about? So in today's class, we started off by summarizing what in the last lecture, the glucose is the most common source of energy and common source. And the microbes, the common microbes, will do their best to extract as much energy out as possible. They take E. coli, for example, if there's oxygen, they will do aerobic respiration to get 38 ATP. And if there's absolutely no oxygen, then they're going to look around to see if there's any other molecule can receive electrons, say nitrate, sulfate, and other biomolecules. And if none of them exist, then E. coli will go into fermentation. Now, yeast is an organism that only does the fermentation. And then we talked about give you examples of other microbes, how they get energy. You have purple sulfur bacteria and cyanobacteria, and then you have hydrogen bacteria, then you have the methanogens, and they both use the hydrogen as the energy source and generates the methane. And then if there's a methanol troops around, and the methanol troops will be very happy because then they can just simply sit there and take out all the methane that's generated by the methanogens, right? So you would think methanogens and methanol troops would they be happy to live together? They will be very happy, right? Because, well, at least the methanol troops will go around and find methanogens to live with methanogens, because methanogens will generate methane for them. All right. So I will stop here and then take questions. Hold on.